Welcome student. Hope you all are happy and fine right now. Now we are going to cover the part number 10 of lesson 6, that is molecular basis of inheritance. Hope you are understanding the last part that I have explained. In today's part, that is part number 10, we are going to understand about the structure and the function of tRNA. In full form it is called transfer RNA, and in another form it is called adapter molecules, which help in the showing a particular kind of arrangement of amino acid and formation a particular kind of protein. So a tRNA help in coding a particular kind of amino acid. So tRNA are called adapter molecules. Remember this. In short form, in fill in the blanks like questions or somewhere in optional type of question, which kind of RNA are called adapters? This kind of question has been asked. In that case, you have to give tRNA. Understood? Next, here I have given the figure of tRNA. Actually, tRNA look like this, and it is a very complicated kind of structure, but scientist has made a very simple clover leaf like structure of this tRNA. In this C, you can able to see there are three main loops, and the first loop, the upper loop contain a particular kind, the 3' end contain a particular kind of amino acid, means the 3' end attached with the amino acid, and here it is a glutamic acid. I will explain all the things. So all these structures are called secondary structures of the tRNA, and this lower structure is called Anticodon loop. Anticodon loop study the code of mRNA. Suppose it is the mRNA, and in the mRNA you can see mRNA means messenger RNA, and it carry the message from the DNA to produce a particular kind of protein. So the mRNA which carry the message has triplet, and each triplet represent a particular kind of code a particular kind of amino acid, and all particular kind of amino acid combine together to form a particular kind of protein, and this particular kind of protein represent a particular kind of characteristics. I am repeating the same thing many times to make you understand. Remember this. So these all are the code, and the tRNA study the code by their anticodon loop. So this loop is called the anticodon loop. Other loops are called secondary loops. So anticodon loop, see here, try to understand. It is G for guanine. Just guanine will pair with cytosine. Similarly, A means adenine. It is a RNA. So in case of RNA, A pair with uracil. Similarly, it is guanine. G for guanine, and guanine pair with cytosine. Hope you have understood. So it is the code. Every code have several messages, single message. So the tRNA read the code, and according to code they carry the protein, and all protein arrange in a manner. Just I want to show you a simple video which make you understand about the working procedure of tRNA. Wait. See. The
30S ribosomal subunit, formylmethionine tRNA, and messenger RNA.
It is a tRNA. Now see how tRNA is get arranging over here. You know that AUG, a codon called AUG, is called starter or initiator. It initiates the protein synthesis. So first of all, the first tRNA which code the first tRNA which code for the first letter or the first codon is AUG, where they will start the protein synthesis. Now see how AUG
The 50S ribosomal subunit then joins the complex. Proteins called initiation factors are also.
So you know that AUG is code for methionine, isn't it? AUG has a dual function. First, it acts like a initiator of protein synthesis, and secondly, it also help in the it also code for a particular protein. So here the particular protein is methionine. So AUG, that is present in the mRNA, code for a particular kind of methionine. And how the methionine is bought? The methionine is bought by transfer RNA. So transfer RNA is responsible for bringing all the amino acid from the cytoplasm and arrange the amino acid in a manner so that it forms a particular kind of protein. See,
but are not shown. The 70S ribosome has two sites to which transfer RNA carrying amino acids can bind. One is called the peptidyl or P site, and the other is called the acceptor or A site.
A site.
There is also a third site called the exit or E site, where transfer RNAs are released. The initiating transfer RNA carrying formylmethionine binds to the P site, a transfer RNA that recognizes the next codon and carries.
Now see how the transfer RNA are carrying. How the transfer RNA are carrying a particular kind of protein or amino acid by reading the particular kind of codon. Means this codon is associated with this. So this tRNA bring this tRNA code with this. Once again, I am repeating. In the anticodon section, see here, it is the codon of mRNA and it is the anticodon section of tRNA. So tRNA study the codon of mRNA and sit over there, and each anticodon bring a particular kind of amino acid and arrange it. See here what happens next.
The second amino acid then moves into the A site. The formylmethionine carried by the transfer RNA in the P site is then joined to the amino acid carried by the transfer RNA that just entered the A site by a peptide bond. The ribosome now advances a distance of one codon, and the transfer RNA that carried the formylmethionine is released at the E site.
Hope you've seen how the tRNA has come. Read how the tRNA has come, read the codon, and according to codon it bring a particular kind of protein and attach the particular kind of protein in chain, and this chain of amino acid make a particular kind of protein. Hope you are understanding.
For RNA carrying the next amino acid now moves into the A site, where the anticodon on the transfer RNA matches the codon on the messenger RNA. The ribosome shifts down by a distance of one codon. As the shift occurs, the two amino acids on the transfer RNA in the P site are transferred to the new amino acid, and the second transfer RNA is released from the E site. The ribosome continues to move along the messenger RNA.
Hope you understand how the tRNA act and function. Means it has a main function to carry the amino acid from the cytoplasm and make a chain during the process of translation. Hope you understood this part. Let's study in detail. So here you can see the structure of tRNA or transfer RNA. The 3' end bind with a particular kind of amino acid It is a glutamic acid, and glutamic acid is caused due to CAG, that is this code, and this code is studied by anticode, just opposite code of the TRNA. Understood? And another part is called secondary loops. Let's study. So it has an anticode loop that has base complementary to the code present in the mRNA. Means it is complementary. Complementary is just opposite code to the mRNA. Understood? And it has an amino acid acceptor. The 3'N is the amino acid acceptor, which bind a particular kind of amino acid. Understood? So tRNA is specific for each amino acid. There are lots of tRNA, 20 kinds of tRNA, and each tRNA have a particular kind of amino acid. Again, each tRNA bind a particular kind of amino acid and bring the amino acid in the formation of protein. Understood? Next, the secondary structure, means the loops in the eastward and westward, is called the secondary structure of tRNA, and depicted means look like, or you can say like seen like a clover leaf. In actual structure, the tRNA is compact molecule. The figure has been given just beside it, and it look like inverted L. So it is the overall structure of tRNA and function at all. Although it has not been explained very well in your book, but I tried to explain it well. Hope for your better understanding. And remember, if you are not understanding, or if you are getting any kind of problem regarding understanding, you can ask me anywhere, anytime. I am always able to help you. Hope for your better learning. Thank you. Make the note, send it. So many students are not sending the homework properly. So first of all, make the note and send it to me. Okay, bye bye, see you.